Hello, everybody. Welcome to Colloquium today. It's my great privilege to introduce to you Chris Lowery. Chris is a paleoceanographer broadly who uses a lot of different things tied together, whether it's paleontology, geochemistry, seismic observations, really brings it all together to understand sort of the response of life to stresses and climate conditions at different times in the past. He did his bachelor's degree at,
University of Mary Washington.
University of Mary Washington. And then after that, he went on to UMass Amherst, where he received both his master's as well as his PhD. And after that, he became the Buckler postdoctoral fellow at UT Austin. And he did such a great job there that UT did not want to let him go. And so therefore, he is now a research scientist at UT Austin, where he's been for the last couple of years. So with that, I've been really excited to work with him. He's been on Expedition 364, so this photo of the rig at Chicxulub crater probably looks very familiar to you. And so I'm really looking forward to hearing him talk about what he's been doing lately with it.
Yeah, thank you. And thank you guys all for having me. I really appreciate the opportunity to be here. You guys have an awesome paleoceanography program, so it's good to come and see everybody and to give you guys this talk. So I'm a IODP Ocean Discovery lecturer next year. So this is kind of like a proto version of what that talk is going to be, with a little bit less like IODP history and a little bit more science, because I'd rather talk about science with you guys. But this is kind of like the kind of first pass version of that talk. So bringing together a lot of stuff I've been working on with Chicxulub, and so really looking at the recovery of life after the asteroid impact. So I applied for this. I was telling some people earlier, right around the time I finished my PhD. I almost didn't apply for the cruise because I had a postdoc already lined up, and I was like, I don't know if we're working on this other project. Should I even apply? Will I be able to go? I was like, whatever. I guess I'll just apply. It's worked out really well. Since you'll see, I've gotten several really good papers out of this, and it's really opened up whole new avenues of research for me. So really great opportunity. Definitely, if you're a graduate student, postdoc, if you have the opportunity to apply for IODP, definitely apply for any cruise you think you might be able to do. Looking at the Chicxulub impact in the crater specifically, we don't have any record of what happened in the Cretaceous. That was all vaporized by the asteroid, but we have a great record of how life recovered afterwards. We have this great marine sequence, which I'll show you, and that's kind of kick-started my interest in how life recovers after mass extinction events, which we're going to talk about here. Before we get too far down the road with that, I just want to acknowledge that a lot of this comes out of Expedition 364, which is a very collaborative effort. All of these people contributed substantially to the science you're about to see. It's a really cool expedition because, I'm a simple micropaleontologist working with people that study impact craters and other planets, the origination of life, all this really cool stuff. It was a lot of fun to be a part of. So the first 2/3 of the talk come from 364. The last third comes from related stuff that I started doing with my friend Andy Frost, who's up here separately, looking at kind of more macroevolutionary things. So why do we care about the recovery of life after mass extinction events? Well, debatably, we're living through one right now. I think it's a question of whether we're truly in a sixth extinction at this moment, but certainly modern rates of extinction are really elevated. We're looking at extinction rates like 100 or 1,000 times above background values. So whether we are currently, already in a sixth extinction or about to enter one, I think it's not really something you can say until you're after the fact looking back. But regardless, we're seeing significant losses of biodiversity, and understanding how the biosphere is going to recover after mass extinction events is really important for understanding the mass extinction that we're in, and kind of putting a frame around why it's important to conserve what we have. So there's canonically 5 mass extinctions in Earth history, major mass extinctions. This goes back to this paper by Sapkowski and Narap back in 1982, where they found the KPG, and then mass extinction at the Triassic boundary, and the Permo-Triassic. Obviously, can't really see it on this plot, but probably the biggest mass extinction in Earth history, the PT, and then one at the end of the Devonian and one at the end of the Silurian, or at the end of the Ordovician rather. These 5 major mass extinction events. This is a more recent plot of origination and extinction from 2008, just to kind of show that a lot of this depends on how you plot it. It depends on the species that you choose and how you treat the data. But however you treat the data, several of these things always stand out, and those are the PT, the Triassic, Jurassic, and the KPG mass extinction events. after mass extinction events, the recovery from mass extinctions, there seems to be this delay after every major mass extinction event in the recovery of life. This is a great paper by James Kirchner and Ann Weil from 2000, showing after the big 5 mass extinctions, a delay in the peak and speciation rates after the event. So kind of the recovery of life is delayed after each of these events. So here's peak extinction, and this gray bar represents the time between the peak extinction and the peak origination. And all of these are about 10 million years-ish, at least on that order of magnitude. This long-term trend. This is a cross-correlation just showing this peak and delay is between 10 and 20. It's about 10 million years is what most people say for this delay in the recovery of life after these mass extinction events. And we see this with higher resolution data too. So this is built on macrofossil, marine macrofossil, macroinvertebrate data at the genus level. And we can look at this as species level too for microfossil groups that we have much higher resolution data for and see the same trends. So this is planktic foraminifera through time. I'm sure here you guys are all familiar with planktic forams, but just in case you're not, they're marine plankton. These protists, the single-celled organisms about the size of a grain of sand. Here's one in the eye of a needle. They originated in the Jurassic, and they kind of like increase in diversity all the way through the Cretaceous. And then here's the KPG. And there's this big drop in diversity right here. And there's a quick rebound, but it's not very much diversity that comes back. And then for the rest of the Paleocene, about 10 million years, diversity is very low. And you don't really start to see recovery to even mid-Cretaceous level diversity levels until around the PETM. And then you don't really get back to close to late Cretaceous levels till the middle Eocene. And obviously most of the Cenozoic seems to be correlated with climate events. The PETM obviously is a big one. Eocene climate optimum, then cooling in the Oligocene, and Miocene events, and the more recent stuff, which is a topic for another talk. But after the KPG, it seems like life took a long time to recover. And we can look at the microfossil record as kind of a proxy for the larger groups that we don't have a good record of, or as good a record of, in Earth past. So obviously the KPG, 75% of all species went extinct, including non-avian dinosaurs, marine animals like ammonites, plesiosaurs, mosasaurs, cool stuff like that, which is, a lot of public interest in this event stems from that. Also, there's an asteroid impact, and that's really cool. And who doesn't love a good, catastrophic story? When we look at microfossil changes across this boundary, we see this across microfossil groups. So these are coccoliths. This is from a paper by Knoll and Follows from a couple years ago. This is a lower resolution data, but here's coccolith diversity at the species level, drops off of the boundary. Lower resolution, but there's a couple bins in here. You can still see this isn't just like here's a data point and then here's a data point. There's still a delay, about 10 million years. You see this in dinoflagellates as well. Diatoms, interestingly, had just really kind of taken off, and they seem to take advantage of this, and then they drop down and then diversify again later. So I think it's very interesting, something maybe that warrants further investigation down the road. But kind of the broad question we can ask of this is why? What controls this delay? You can kind of think about this in two separate ways. One of them is this delay is caused by environmental effects. There's some sort of environmental after effects of mass extinction events that hold back recovery of life. And this, I think, makes a lot of sense when you think about like the big volcanic-driven extinction events like the Permo-Triassic. You have these huge volcanic eruptions that really perturb the climate system for a long period of time. Maybe that holds things down. But we also see this after the KPG, which was caused by an asteroid impact, which was very, very fast environmentally, right? The asteroid hit, you have a couple years of global darkness, cooling, shutdown of photosynthesis, but then all that stuff gets removed from the atmosphere and the environment comes back very quickly, and yet we still see this long 10 million year delay. So if we can study the reasons for this delay after the KPG, maybe this will help us understand why this delay occurs after other extinction events as well. So whether it's environmental or the other hypothesis here is that this is actually driven by broader macroevolutionary processes, that this is just some sort of fundamental speed limit in the diversification of life that's always going to happen. Whenever you eliminate a whole bunch of diversity, this is just how long it's going to take to come back. In which case, after the modern mass extinction event, if we really do go through a sixth extinction, or even if just modern levels of diversity loss, like we've definitely gone through a mass extinction already. We're currently experiencing a mass extinction. It's not sixth mass extinction levels, like 70% of the biosphere hasn't gone extinct yet. But we are definitely experiencing elevated diversity loss. And how long is that going to take to recover after, if we stopped right now overfishing, stop polluting the atmosphere, stop CO2 emissions, how long would it take biodiversity to recover to pre-extinction values. This would suggest that it would take a very long time if it was macroevolutionary based. So we can look at the KPG to try to understand this. So KPG, here's a great boundary section, very famous boundary section in Gubbio Gorge in Italy. Here's Simone Galeotti for scale. This is the Cretaceous, this is the Paleocene. As you can tell, it's a famous section because everybody's taken a little chunk of rock from the basal Paleocene here. And from a microfossil perspective, we've always known this is a pretty fast extinction event. These are plankton foraminifera both before and after the KPG. This is from a ODP site off the coast of Florida, but just kind of superimposed on these Italian hard limestones. But you can see, late Cretaceous plankton forams were large, they were diverse, they were very complex. They filled very specific niche spaces in the environment. They did a really good job at one particular thing. These exist in these rocks all the way up to this level, and then they disappear. And then in the very next layer of limestone after this clay layer, you find just a few species, very simple morphologies. They're all small. They have very broad, generalist life strategies. And this is, all the way up to this level, you see these things, and they disappear. It's a very fast event. Suspiciously fast, people used to think. And so when people were studying this back in the late 70s, these two guys came out here to this outcrop. This is why this is famous. This is Lewis and Walter Alvarez. And they thought maybe there was, it was so fast, maybe it's because there was missing time here. There's a bit of a gap in the depositional record. So they looked at an element called iridium, which is not found naturally in the Earth's crust, but is very common in space rocks, in meteorites, which means that there's a very slow flux of it into the oceans, because every time there's like a shooting star, every time a little piece of dust burns up in the atmosphere, that settles out a very little bit of iridium that lands in marine sediments, and it accumulates at a set rate that we know. And so if we look at this abundance of iridium through these sediments, if there was a little unconformity there, if there's a little condensed layer, we'd expect to see a little increase in the amount of iridium and then to go back to background rates. And that would say, oh, hey, there's an unconformity here. And that's what they were looking for. And what they found was, instead of just a little blip, they found this huge increase in iridium. It's like 100 times background. I forget exactly the order of magnitude, but it's more than you'd explain with 10 million years of missing time. And they're like, oh, crap, maybe this is actually an asteroid impact, and that's where that hypothesis came from. A quick aside, because I like this story. This is my colleague Jan Schmidt. I'll go through every single laser pointer. My colleague Jan Schmidt, he was working on the same thing at the same time in Spain, looking at iridium abundances across the KPG boundary. He sent his samples off to the lab. They called him up a couple months later and said, Jan, there's a problem. Your samples are contaminated. We're really sorry. Can you send us some more? We'll run them again. And he was like, oh, yeah, whatever, fine, okay. Send him the samples. And while he was doing that, these guys published their paper saying, look at this huge abundance of iridium. It's an asteroid impact. And Jan was like, ah, God. So he got scooped, basically because the lab was, misinterpreted. They didn't tell him what the anomaly was, what the contamination was. So he published his paper a couple months after the Alvarez's published theirs, which was kind of a really 1-2 thing that kind of helped push the community in this direction. It was a very controversial hypothesis to start out with. At the end of the day, like, these guys they're friends now, Walter and Jan. They have vacation homes together in the Alps, or in the Apennines in Italy. Anyway, so in addition to the iridium layer, there's a lot of other stuff that marks the boundary. There's spherules, or tektites as we call them. This is from a newer section that's been discovered in Gorgonilla Island in Colombia. These nice glassy spherules, these are vaporized impact rocks that have condensed in the atmosphere and fallen out. A lot of them are round, a lot of them have dumbbell or teardrop morphologies. Some of them have bubbles in them in the middle, which is cool. People thought, oh great, there's this little bubble here that's formed in the atmosphere. We can sample this and study the Cretaceous atmosphere. And they crunch these up underwater to get the gas out. And it turns out there's no gas in there, it was a vacuum, which means that these things crystallized in space. And this little bubble is a little bubble of Cretaceous space that then got preserved inside of this spherule and then preserved on the ocean floor. I'm really trying hard not to make this whole talk just cool factoids about the KPG. It's a struggle. I'm just throwing out a couple things, but this stuff like that is really cool. So this is a conceptual model of what happens at the KPG boundary. This is from that big review paper by Schulte et al. In Science about 10 years ago now. So these are the range of different species of the boundary. A lot of things go right up to the boundary. Everything we have a good record of, basically, goes right up to the boundary and goes extinct at the same level at the boundary. A few species that were rare in the Cretaceous make it through the boundary, bloom for a little while, become really abundant, and then go away and fade back to background things. And then a lot of other species that were present in the Cretaceous begin to diversify and set up the rest of the Cenozoic diversity of life. This is exactly coincident with big changes in ocean chemistry, big drop, negative excursion in carbon isotopes, big drop in calcite weight percent, and this lines up exactly with the iridium spike, showing that it does line up very nicely with the KPG boundary. IODP was really instrumental in studying the impact hypothesis. These first papers about the impact hypothesis were from terrestrial sections from outcrops. And then everybody went back to all the DSTP cores that had already been drilled by this point in time, looking for more evidence of the boundary. So this is from a paper that came out in that special issue of Oceanography a couple years ago, or a couple of weeks ago, sorry, time flies, I tell you, showing all the different KPG boundary sites that have been drilled by the Ocean Drilling Program. And this is the number of citations by site, just showing that different ones. But this is a huge amount of papers from some of these places. Most of our knowledge of the KPG, of the effects of on life of the Chicxulub impact, come from marine sections cored by IODP. And this has kind of led to some a lot of data, which is great, but also a lot of data which leads to kind of some conflicting views sometimes. This is a great paper by Jiang et al., one of Tim Brailauer's students, looking at the recovery of nanoplankton diversity after the KPG, looking at hemispheric changes. So a bunch of sites in the Southern Ocean and a bunch of sites in the Tethys and in the Pacific. And they found this amount of species that goes extinct are much higher. More species go extinct in the Northern Hemisphere than in the Southern Hemisphere. You can see this in the same plot here too. This is just diversity over time at different sites in the Northern Hemisphere, in the Southern Ocean, and the South Atlantic. Which suggests there's some sort of geographic control on extinction and recovery, and that they suggested that the Northern Hemisphere was hit harder by the impact, and that something in the Northern Hemisphere was holding back the recovery of nanoplankton. They proposed that toxic metals from the asteroid impact built up in the oceans and kind of, particularly in the Northern Hemisphere oceans, and held back the recovery of life up there. We can also see this somewhat in records of Paleo Productivity. So this is a bunch of biogenic barium records, barium mostly, but also other proxies. So that the black here are places where you see a decrease in productivity across the boundary. The gray are places with no change, and the white are places that actually see an increase in productivity across the boundary, counterintuitively. Globally, productivity dropped on balance, but in some places you actually see an increase in productivity. And what we see in a broad sense is that in the Northern Hemisphere, particularly the North Atlantic, around the Gulf of Mexico, the Tethys, and North Atlantic, closer to the impact crater, there was a stronger drop in productivity, again, which suggests a geographic trend of slower recovery or worse effects closer to the crater, which again would suggest some sort of environmental or impact-driven driver holding back productivity, or not productivity specifically, but just more broadly the recovery of life after the KPG. So is there a geographic trend to recovery? If so, it should be worst in the crater, right? That's a pretty, like if you're going to imagine the place where all these things should be the worst, probably ground zero would be the place to look. Where is the crater? We haven't talked about that yet. Excuse me. When the hypothesis that an asteroid impact caused the KPG mass extinction was first proposed, one of the big criticisms was, like, all right, smart guys, if it was an impact, where's the crater? And for a long time, they didn't know. There's actually a lot of thought that maybe the crater just wasn't going to be found, that it landed in the ocean, in deep water, and didn't form a crater. Landed in some place that was subducted or that became mountains or otherwise was destroyed by the last 66 million years of plate tectonic action. But as people started to study, particularly with KPG, with the ocean drilling sites, what we now know are places that were distal from the crater. You have a very thin boundary layer, millimeters thick. Everything's in one little clay layer with the iridium spike and the ejecta and the shocked quartz and everything else. Here's the iridium spike, by the way. Here's a piece of shocked quartz, and then you get the spherules I showed before. As you get a bit closer to intermediate distances, the boundary layer is centimeters thick. You get a layer of spherules with a layer of finer grained clay layer of ejecta on top, again with the iridium spike within that clay layer. Even closer to the crater, you find spherules and then tsunami deposits, high energy clastic deposits, and then with the clay layer on top of that with the iridium spike, sometimes multiple iridium spikes, as continued tsunami action kind of reworks things. And then really close to the crater, meters, tens of meters thick, you get a big graded clastic, ground mass flows from the impact itself, and then, kind of differentiated stuff, again with the clay layer on top. So people started kind of looking around the Caribbean area for crater. In 1991, Alan Hildebrand and others were looking at gravity anomaly maps, found this very suggestive looking round thing on the Yucatan Peninsula. and went back to some old Mexican petroleum company cores, Pemex, petroleum company cores from the 50s, where they'd also found a suggestive looking round thing, and they thought, aha, there must be oil in that because it's a structure. So they drilled a bunch of holes. There's no oil in it. They put the cores on a shelf in a warehouse that actually burned down, and then there's a couple other cores on another shelf in another warehouse. They're able to go, Hildebrand and others were able to go and look at those cores and look at the actual lithology there, and they found impact melt rock, and they found shocked quartz, and they found other markers of an actual impact crater. So they wrote this paper in 1991, basically describing the existence of the crater. So that was 1991 was when they found the crater. We drilled, the first IODP proposal to drill offshore was 1996. And then we drilled it finally for the first time in 2016. So a little bit longer than most core proposals going through the system. This is a magnetic anomaly map, and I'm sure you've heard about this from Sonia too, but I love this stuff, particularly because, again, simple micropaleontologist, I don't get to talk about space that much. But this is the only crater on Earth that has a peak ring. Peak ring, of course, is a discontinuous set of hills on the inside of larger impact craters. Chicxulub is the only one with a clear one on Earth. And so if we want to understand how peak rings form, we can go to Chicxulub or we can go to the moon. And there's probably no foraminifera on the moon, so I'm glad we decided to go to Chicxulub instead. We wanted to drill offshore because we could get this beautiful seismic data. We could actually see what we were doing. There's been several cores drilled onshore, and they were drilling not quite blind, but I mean, obviously, it's a karst environment out there. You have it's all limestone with a lot of karst in it, so you really don't get nice seismic data onshore. And so to get the peak ring, we really wanted to go offshore and see this beautiful seismic packages to see exactly what you're doing. So there's these surveys that were shot in the late 90s and early 2000s. This is this line R3 right here, and the star is our drill site. Here's our drill site right here. You can see we drilled through post-impact sediments into the peak ring. This nice low velocity zone is right at the top of the peak ring. We had a bunch of different objectives. The one that I was particularly focused on is how does life recover afterwards. And again, just because I think this is really cool and exciting, right before we drilled, there was a paper that came out summarizing the two hypotheses for how peak rings form in impact craters. And I'm not going to go into much detail at all about this, but basically in one of them, the asteroid hits, forms a bunch of melt. That melt absorbs a lot of the energy, and most of the energy then goes out, and you have this big, all this stuff gets pushed out, and then this collapses back in, and the outer stuff collapsing back in kind of runs up against the central uplift, and that forms the peak ring. In which case, we'd predict that the peak ring is formed of shallow crustal rocks, probably Cretaceous carbonates. And the other model, there's less melt formed. It digs out a deeper transient crater. Everything gets rebounded, and the stuff on the sides kind of flops out and then falls back over, and that's what forms the peak ring in this model, in which case we'd expect to find deeper crustal rocks, probably some sort of crystalline basement. So in the year 2016, you can go out and drill a hole in the ground and answer a fundamental question about basic processes in the solar system just by seeing what kind of rock is at the bottom of that hole, which I think is really cool. I mean, we think about science as being really complicated, and it is in a lot of ways, especially this stuff. But just a simple hypothesis test of let's go drill into the peak ring and see what kind of rock it's made of. I just think is really cool that that's the kind of thing that we can do these days. Still. So this is IODP. Normally we'd use the JR. We can't because the water here is too shallow. Jim, if you want to use this as the logo for the next meeting, feel free. Yeah, so 17 meters deep, way too shallow to use a drill ship. So instead we used a lift boat, the Liftboat Myrtle. Which is a great name, I think, for a lift boat operating out of Louisiana. Basically a barge with feet. It's got these long legs that it can kind of lower down in the water and raise it up out of the water, produces a stable platform. We got a land-based mining drilling rig we welded to the front of the ship and then drilled with that. Excellent recovery, basically 100% recovery through the whole hole, which is awesome. The water was so shallow that when we got bored, tied a GoPro to a rope and lowered it over the side. We called it fish cam. Here's a fish. This is a perspective. This isn't a huge fish, but this is the foot, one of the feet of the thing. So this is really shallow water. Not a normal cruise in any sort of sense. We didn't stay on the ship. I was offshore for 2 weeks as a biostratigrapher. So as we were drilling through those post-impact sediments, I was offshore doing that work, working a 12-hour shift. Once we got into the impact melt rocks, I was completely out of my depth. Pun not intended. But as soon as we drilled down to that stuff, I was gone, and we brought out impact petrologists to look at that. For the 2 days, while we were getting that shift together, we were looking at like hydrothermal minerals, and me and Tim were like, Tim Bralauer, the nanofossil worker on the ship, we're doing like hardness tests. We're like, all right, does it scratch glass? What kind of mineral is this? We're like, get out of here, guys. We'll do somebody different. The platform didn't move. We all came out on crew boats. So you could get on this boat, and then they lower a basket down, you grab onto the basket, and they raise you up, which is a lot of fun. Nice view of the ship as you're getting on. Here's a closer view of the whole thing with some people for scale. This whole white structure is basically the living quarters. The galley's on the first deck. This is all living quarters and common space, but right outside of living quarters, so you have to be quiet. There's really no place to like sit and talk other than the galley. And then up here's the bridge. And then everything forward of that was drilling. We had a bunch of air-conditioned shipping containers that were made up all the labs of the Myrtle, and then we had the drill rig out front. And here's a view from the basket looking down as I came onto the rig, looking at. So here's the rig itself, the drill floor here that's kind of cantilevered over the front, and you're looking straight down into the Gulf of Mexico. Pretty cool. So what do we find? There were 600 meters of post-impact sediments. Here's the very base of that section. Very base of this. This is earliest Paleocene. Right here, this paper is still, I don't think they've actually written it yet, but they're still kind of working on this PGE, this Iridium and other platinum group element story. But they've talked about it in conferences, so I don't mind telling you guys that there's a big enrichment of Iridium and other platinum group elements right here at the top. This is that big Iridium spike that marks the top of the boundary. And there's this 80 centimeter thick brown micritic limestone we call the transitional unit. We interpret this as settling layer, basically. This overlies 130 meters of impact breccia that basically is the stuff that fell back into the crater after the crater formed. At some point in there, water rushed back in. You have a tsunami and then multiple seiche waves kind of sloshing around in the basin for a while. And when all that settled down, you had very, very turbid water. And so that stuff is going to slowly settle out of suspension, and that's what this is, this brown settling layer, we call it. The fact that the iridium anomalies at the top of that tells us that this whole thing fell out of suspension within a couple of years. Because we know that the dust would have settled out of the atmosphere a couple years after the impact. The fact that that occurs up here tells us that this is less than a couple years after the impact. Below that is this breccia. Up here, it's sand-sized. You can kind of see some cross-laminations. You see a little bit better in the CT data here. This is from the tsunami deposit. This is basically 100-some meters of coarsening downward breccia. This is kind of gross grain size. You go from fine sand to coarse sand to small pebbles to cobbles to larger cobbles that fill up the whole core barrel down to the base. This is a plot of maximum class size kind of going all the way up through this section. So basically just kind of coarsening downward the whole way. That in turn overlies impact melt rock. Here's the base of that breccia, and then there's impact melt rock, and then below that is granite. Which is very exciting. I wasn't on the ship anymore when that came up, but everybody's really excited because you'll recall that granite means that hypothesis B about how peak rings are formed is the correct hypothesis. So they actually started writing that paper on the ship before they even split the cores. So this is the whole section. So we've got a lot of post-impact stuff, and then impact breccia, melt rock, and then granite all the way down. And again, just because I think this is really cool. This is an animation showing the crater formation process that they were able to make based on the data we found on the ship that came out with the paper that they wrote while they were still on the ship that got published in Science in 2016. This is the process of crater formation. So to play this again, a little sneeze there at the end. Let's see. Give me back my thing. So what this is the blue rock here is what eventually becomes the peak ring. And this whole animation is sped up, but it's the first 10 minutes after impact. So you get a transient crater that's 30 kilometers deep. Everything gets vaporized, that gets put up in the atmosphere. You have a transient peak that's 20 kilometers high, and that falls back down. That's all about 5 minutes, by the way. It falls back down, kind of settles out, and the peak ring kind of comes out and surges out, and then stops there at the end, and there's the peak ring, and that's where we drilled. At some point after that, minutes, hours maybe after that, the tsunami comes back in, the water comes back into the basin, and then you get this settling layer starting to come out of suspension. Within this settling layer, we find evidence for life. And remember, we found, this is where the iridium anomaly is, so this is within a couple years of the impact. And we actually find life within the settling layer. This is a paper that was led by me that was published last year. showing this rapid recovery of life. We find right about here we start seeing burrows, which to me is the strongest evidence for life. There's some burrows here. They never show up well on projection screens. They're beautiful in the crater. They just don't never photograph quite right. But you can see some Planolytes, chondrites burrows up here, beginning about this level right here. And the beautiful thing about burrows is they can't be reworked from the Cretaceous. They're primary sedimentary features, right? So these aren't Cretaceous burrows that got washed back up. They're also not coming down from above because they're not filled in with post-impact limestone like we know we'd find. There's a couple burrows at the very top that are filled in with post-impact limestone that are also filled with post-impact, Danian planktic foraminifera. We can very easily spot those. Whereas down here, these are formed of the same stuff that the rest of the transitional unit is made of, which means these are formed at the same time. We also see evidence for this in microfossil data. So these are planktic foraminifera. This is foraminifera per gram of sediment. And then this is the percentage of survivor species of planktic foraminifera. So almost all species of forams went extinct. A few survived. They were not very common in the Cretaceous. And in this lower section of the transitional unit, there's some winnowed surfaces or little layers here that were probably deposited by the waning energy of these seiches going back and forth. And really high abundance of foraminifera, and all of them are Cretaceous reworked forams. There's a few species that became survivor species, but I don't think these were not survivors. I think these are truly reworked Cretaceous. And you see, along with that, lots of these big, unique, really specialized Cretaceous species, ranging in age from the Campanian to the Maastrichtian. So a big jumble of ages of Cretaceous species right down here. That abundance really drops off pretty quickly and stays low the rest of the way through the crater. And as you get through the transitional unit, I beg your pardon, and as you get further up section here, you start to see an increase in the number of foraminifera per gram, and that is associated with an increase in the percentage of survivors. And that starts right around in here, right about the point we start seeing these burrows coming in, about 55 centimeters within the depth of this core section. We see this increase in the number of foraminifera along with a percentage of survivors. There are fewer data points here on the forams per gram side than there are over here because I had an undergraduate washing some of these samples for me and she forgot to weigh a few of them, which means you can't get the weight per gram. Anyway, but it lines up. It's okay. It's got published in Nature, so it worked out. Anyway, but you see like this increase in the forams per gram and the survivors kind of increases through here, which suggests to me that these are not reworked Cretaceous things. These are true survivors living within the crater. This is the same time we see these burrows. With nanofossils, we see similar trends. This is work done by my colleague Tim Braylauer, showing an increase in the percentage of the species Micula, survivor species, up towards the top of the section. Nanofossils, as I mentioned, are phytoplankton. They're an order of magnitude smaller than forams. I love this picture. Here's a foram, and then here's some pores on the foram. There's a nanofossil right there. You can't see them with your naked eye, but as everybody likes to point out, when they bloom, you can see them from space. Here's a bloom of Milion huxleyi in the English Channel. We look at these nanofossil assemblages within the transitional unit. Nanofossils are all really, really small. You wouldn't expect them to be really strongly size sorted. Like maybe you could make the argument that because the survivor species of forams are smaller than the ones that went extinct in the Cretaceous, maybe what we're seeing there isn't true survivors. Maybe those are just size sorted. You get the smaller survivor species up towards the top. You wouldn't expect to see that with the nanos, and so we look at these. These are the Chicxulub populations plotted against latest Cretaceous populations from a bunch of other sites. What we see is anomalously high abundances of certain survivor species in Chicxulub. Reticapsa, for example, is way more common at Chicxulub than anywhere in the Cretaceous. Watsonarea, too, really common. And we see very low abundances of other common Cretaceous species within this assemblage that we interpret to be survivors. So really low abundances of Prediscosphere and Ipholithus, Chiascozygus, these other common Cretaceous nanofossils. And this was possible, this rapid repopulation, or population, because I guess there was no crater to have a population before. Anyway, this rapid population of the crater is possible because the crater itself was open to the northeast. It wasn't a fully enclosed basin like the Chesapeake Bay impact crater which was, the Chesapeake Bay crater was totally closed off, and so you had this anoxic layer that formed and kind of delayed the recovery of life there. So this is actually faster than the order of magnitude smaller Chesapeake Bay impact crater recovery of life-wise. So the whole thing is open at depth, so you have lots of Gulf of Mexico water coming into the crater, circulating through. And so life appeared within the crater within years because the crater was habitable within years. So even at ground zero of this mass extinction causing impact, you could have life living within a few years of the impact. This is environmentally just a very quick event. So what about longer term? Sticking with nanoplankton for a second, this is work by my colleague Heather Jones that's in revision with geology. Hopefully will be resubmitted soon. This is abundances of different species of calcareous nanoplankton in the crater and in other sites. So the gray here is the Chicxulub crater. The green is Ferrata, the Ferrata section and the Tethys, and the black dashed line is site 1210, which is Shatsky Rise. And the calcareous nanoplankton after the impact had a lot of big blooms of what they call disaster species, right? These species that did really well in this kind of chaotic post-impact world that were really well adapted to it. They bloomed, they did great. And then as those species kind of waned, other species came in, and you see successive blooms of different species coming through. They call these boom-bust cycles, Heather calls them. And so basically as the environment changes from disaster to kind of post-disaster, it favors different species which briefly bloom and then go away, and then another species briefly blooms and then goes away for another and another. And eventually you get back those booms all in, and you just have a diverse nanoplankton assemblage like you have in the normal ocean. Well, what we see at Chicxulub is that this process takes a lot longer than it does elsewhere. It's actually about 1 million years before these boom-bust cycles start to kick off. And these aren't lined up the same everywhere. So at 1210, you kind of see these starting hundreds of thousands of years after the boundary. At Ferrata too, but the timing of these peaks kind of don't quite line up from site to site. But at Chicxulub, in the crater, these things take a long time, which suggests maybe that there is some sort of environmental effect here. So we can look at this environmental stuff in a bit more detail. This is a paper or not a paper yet, this is a manuscript by me and a bunch of other people that's going to be submitted as soon as Heather's paper is accepted, where we're looking at more broadly the paleoceanography of the crater for a couple million years. And so there's a lot here, so let me walk you through it. These are age on the side, so this is 66 million to 63 million, so this is, we're zooming out a bit here looking at the whole lower Paleocene. These are planktic foraminifera by depth habitat, so species that occupy the mixed layer versus the thermocline versus the subthermocline. Planktic foraminifera by trophic strategy, so kind of broadly grouping them into eutrophic and oligotrophic species. Heather's nanoplankton data, accordion down into one particular column. These are foraminifera-sized calcospheres, so nano-sized calcospheres bloom all over the world in the early Paleocene. And we started to see some foraminifera-sized ones up here too, which just means they're getting bigger, probably. Then benthic foraminifera, percentage of benthic foraminifera, which should here respond to the flux of organic matter from above. Barium-titanium ratio, which is a proxy for export productivity. Titanium-aluminum ratio, which is a proxy for fluvial input into the broader Gulf of Mexico. TOC, and carbon isotopes, which is a lot. And there's, I plotted all this data together and spent like 2 months just kind of staring at it, rubbing my temples and being like, what's the story I can tell with all this stuff? But broadly what we see, and there's a lot going on here, this is all numbered because there's a lot of smaller trends, but I just want to focus here on the larger picture. And on the broad scale, what we see is high productivity in the crater from the get-go. The very earliest Paleocene, the earliest post-impact sediments have high productivity. That high productivity lasts about, highest productivity lasts about 300, 400,000 years and starts to drop off, but it's still high through about 1 million years after the impact, and it finally bottoms out up here. And we can see this with export productivity with barium titanium, and we see it with benthic foraminifera, which again are dependent on the amount of food coming to them from above, among other things which we can rule out. I don't want to get into that right now, but you can ask me about it later if you'd like. but high productivity early in the Paleocene, declining after about 1 million years. That's lined up with the change from these two survivor species, Cerviciella, which is the new name apparently for Thoracosphaera, which is that calcosphere that everybody sees blooming in the early Paleocene, and Barutosphaera. These are the two disaster species that bloomed and did really great in the earliest Paleocene here. They start to give way to these boom-bust cycles, starting to diversify the nanofossil population around the same time that the productivity bottoms out. And this all lines up with changes in water column structure, so looking at mixed layer versus thermocline versus subthermocline foraminifera, there's some variation down here, but broadly there's more mixed layer habitat down here, which suggests a weaker, less well stratified water column. About 1 million years afterwards, you start to see establishment of a really strong subthermocline and thermocline habitat for these things to live in, suggests an increased stratification. And this change occurs along with a broad, longer-term increase in the amount of the titanium aluminum ratio, which should correspond to the amount of fluvial material that's being transported into the Gulf of Mexico. So to synthesize all of that, the changes in the nanofossil population are being driven by changes in productivity. This is Heather's hypothesis that this data supports, that changes in the nanofossil population, as long as the productivity is high, these survivor species are doing well. They're happy, there's nothing to change the environment. As productivity, as nutrient abundance starts to decline, these things start doing less well, and other species can kind of start to elbow in to the ecosystem, and you start to see these boom-bust cycles start. And we see that here with this productivity signal, and we think that this productivity change is driven by changes in water column structure. So you have a well-mixed water column, you have a lot of nutrients kind of getting circulated up into the mixed layer. As you start to get more stratification going on, you lose that source of deeper nutrients upwelling. And so a more stratified water column is a less productive one here. And that stratification, we think, is driven by the flux of basically fresh water into the Gulf of Mexico basin. So more fresh water, more stratified waters, which leads to lower productivity, which leads to diversification and change in the nanofossil population. So,
what would cause that influx of fresh water?
Good question. Yeah, so we would like run out like several links down the chain, but that would be a good like next step question to kind of get at. But I think, but more broadly, just like North American or Northern Hemisphere or regional hydroclimate, maybe land use changes. You maybe make a prediction that recovery of terrestrial populations of plants might drive changes in this. Although 1 million years is well after the forests would have recovered. Anyway, so that's kind of broadly what's driving that. So it's not that within the crater, it's not that this delay in the recovery of nanofossils is not driven by the crater-specific processes themselves. They're just driven by more broad regional oceanography and climate is the main point here. And so, again, more broadly, we can say that the recovery of diversity is not driven by environmental processes. There's no lingering environmental effects in the crater. There are no like enrichment in toxic metals or anything like that. What's driving this is something else. And so what that something else might be is what I want to talk about for the last couple minutes here. So some sort of larger scale, broad macroevolutionary process. And I apologize because we're going to take a sharp left turn into paleobiology for a second and talk about ecospace. So that paper that showed really nicely that 10 million year delay in the recovery of ecosystems after all the major mass extinctions by Kirshner and Weil also proposed a cause for this delay. And they suggested that it's basically the amount of time it takes to rebuild niche space after these mass extinction events. So niche space, or maybe more appropriately now, we would probably call this ecospace, but you're kind of familiar with the concept of niche space, right? Like it's the particular way that an organism fits into its ecosystem. This is a super complicated plot that nevertheless is one of the best ways I've found to visualize this. This is showing ecospace occupation in the Ediacaran, and then the Cambrian, and then the recent. And these are just different boxes, basically, that organisms might occupy, niches, so to speak. So this is, you can't read this, you're not supposed to, but this is like pelagic. These are erect and superficial and semi-infaunal and infaunal and deep infaunal. This axis is suspension feeders versus superficial deposit feeders versus mining feeders, grazing, predatory, etc. And then this axis back here is attached versus unattached, and if it's unattached, because it moved fast or slow. So all of that kind of sums up different niches that organisms might occupy within an ecosystem. And so when you have an extinction event, you don't just eliminate species, you also eliminate the niches that they occupy. We think about niches kind of like this, the way these people think about niches, as like specific like slots that organisms can fit into. But that's not quite how things work. It's not really like a parking space you can just kind of pull a car in, and that's the niche that you occupy. For example, if you occupy like an infaunal niche, you need a reason to do that. Why would an organism live below the seafloor, right? It's got to be pressured to for some reason. It's got to have like a predator it's escaping. It's got to have a food source down there or something along those lines. And so it's not just the fact that a seafloor exists that you could live underneath if you wanted to. You need a reason to do that. And that's based on interactions of organisms within an ecosystem. So when you wipe out a bunch of organisms, you wipe out those interactions and those niches disappear. An example I like to use is like plesiosaurs. After the KPG, plesiosaurs went extinct, right? Plesiosaurs, a lot of them ate ammonites. And so plesiosaurs go extinct, then the ammonite eating niche is open. Except ammonites also went extinct, so that niche just disappeared. And so these niches are kind of the scaffolding on which diversity is built. And so you need to have these niches and reconstruct these niches before you can have the diversity come back. You can kind of see this qualitatively with foraminifera. With foraminifera, instead of ecospace, the strategy of these things living in particular niches, we can kind of extrapolate that into morphospace, or the different shapes that the foraminifera take on. Basically, if we assume that the shape of foraminifera has some sort of function with its life habit, then we can assume that the variety of shapes, the level of complexity of the shapes, has something to do with the amount of ecospace, the amount of niches that these organisms occupy, right? So in the Cretaceous, foraminifera are really diverse. They're also very complex. A lot of weird, cool shapes going on in the Cretaceous, which suggests that they occupied a large amount of morphospace. They had a lot of different niches that they occupied. You have a big extinction event, all of that stuff goes away. There's 3 main survivor species. They all have very simple morphologies. They occupy very broad niches. They're very simple. There's much less ecospace or morphospace, which they are taking up. You start to have the recovery in the early Paleocene, just a couple 100,000 years after the boundary. A lot of species come back, but they're all still pretty simple. These kind of simple forms. They're not occupying a whole lot of morphospace. And it's not until the very end of the Paleocene, about 10 million years later, you start to see these really bizarre and complex forms start to come back, which show that they've occupied a large amount of morphospace. They're occupying a lot more niches, and those niches allow them to diversify and fill out. We can look at this quantitatively as well. This is a paper that I just published a couple weeks ago. My colleague Andy Frost measured a whole bunch of different attributes. This is just basically to show that with foraminifera, there's a bunch of weird stuff we can measure to kind of quantify the shape of the shell. We picked 10 that we thought did the best job, and we plotted it up over time. So each of these gray bars is the range of a foram species here on the x-axis, and its position on the y-axis is how complex it is, based on a bunch of different measurements of its morphology. And you can see there's a big range in the Cretaceous, but the mean and the median, pretty high. Big drop-off in complexity, as you can see qualitatively from the last slide. And the early Paleocene, if you look at the mean or the median, it kind of varies of whether it starts to recover right away or it actually goes down a little bit, comes back up. But you see that recovery of complexity coming back up here until right before the PETM, when diversity finally comes back, which suggests that this is really what's controlling the recovery. But is it controlling the recovery, or is it just a result of the more species you have, the more complex things are? So we can kind of think about this, look at this a bit more detail. And for this next bit, you just need to understand a little bit about Aquariumtrophic strategies. So in the early Paleocene, there were non-spinose foraminifera. They had, their pseudopodia, their tentacles. They float around. They could eat stuff, but they didn't have any structure to their tentacles. And so whenever something that could swim got ensnared in their little web, it could just kick and spin and get away. In the earliest Paleocene, they evolved spines. Some species did. And the spines provided a solid structure on which those pseudopodia could grab onto things and hold onto them, which allows them to actually eat multicellular organisms, copepods and brine shrimp and all this stuff, which is really cool. Right? This is a protist. This is a single-cell organism that can eat arthropods. It's really punching up evolutionarily. It's very impressive to me that they can do this. And then finally, in the middle of the Paleocene, some of them evolved symbionts, which is basically a strategy to let them do better in oligotrophic or low productivity waters. So they have these dinoflagellates or algal symbionts that live outside the shell. When they're ready to reproduce, they pull those in and they eat them, and they use the energy to reproduce. So different trophic strategies. These two evolved in the Paleocene. Some Cretaceous things had symbionts too, but all of those went extinct at the end of the Cretaceous, so this had to re-evolve, come back. So Andy and I split this out as part of the work we were doing, and we looked at a couple different things. We looked at the mean complexity, that got cut off a little bit unfortunately, but a mean complexity over diversity, basically showing how quickly these things fill out. And what we'd predict is that if you add species one at a time, becoming a little bit more complex each time, you'd basically see a straight line. So like a one-to-one change should have a line with basically no slope, which is what we see in the Cretaceous. Even though there were a ton of species and they're all really complex, the rate of change was very low. In the early Paleocene, there's a big jump. So this is, the black is the mean complexity over diversity. The blue is just for spinos, and the green is just for symbiont bearing. You can see there's a big jump in the basal Paleocene, and you see these other jumps in the middle Paleocene, these kind of elevated rates. And those are associated with these new evolutionary innovations. So the mean jumps, and you can see this just both with the evolution of spines and the non-spinos things, the carryovers from the Cretaceous, diversifying to refill the niches that were available to them. And then the spinos things opening up new niches, these big jumps in complexity of their shape, showing that they're occupying a whole lot of more morphospace, showing they're occupying a lot more ecospace. So they're opening up a lot of new niches that can be occupied. Same thing with the symbiont-bearing ones. When they come on the scene, really high rates of complexity over diversity, again showing that they are opening up new niche space that then is being filled in later. And these big pulses kind of last through the Paleocene. The spinos ones, there's another jump in spinos in the Eocene, but the mean rate is only elevated in the Paleocene. So you have this interval in the Paleocene where you're really rapidly rebuilding ecospace as these things start to get a lot more complex as you open up new niches. And that here we think is what's controlling this long-term diversification. I want to leave some time for questions, so I'll leave this part out. We can talk about it later if somebody has a question. But basically, the initial recovery of life in the 30,000 years after the impact filled the available ecospace, and then the rest of the recovery of life was paced by the need to build new ecospace that things could then occupy, to rebuild those niches, those ecological interactions that drive the diversity of life. What about climate? Again, I want to leave a little bit of time for talk, for questions. So I'll just say that climate, we looked at it and it doesn't affect anything. Ask me about it later. This paper was published a couple weeks ago. It got picked up a little bit in the press, which is always cool. I like this paper or this article because they just used a bunch of different pictures of piles of skulls to talk about our work, which is exciting. Anyway, the takeaway here is that this 10 million year delay in the recovery of life is driven by fundamental evolutionary processes and not some sort of extinction-specific mechanism holding things down from some environmental change. This is just every time you have a big loss of diversity, there's a fundamental speed limit, and this is how long it's going to take to recover, which means that the current extinction event that we're living through is also going to see this level of delay in the recovery of biodiversity in the future, which is really kind of terrifying. You think, like, obviously everything is longer than human timescales, but if we stopped emitting CO2 right now, most of the CO2 would be out of the atmosphere in thousands of years, maybe 10,000 years, like that order of magnitude, right? The climate system, because there's a lot of feedbacks, would probably be out of whack for a bit longer than that. But what this suggests is that with the extinctions that we're causing now, the biosphere is going to be affected for 10 million years, at least, which is really humbling, and which also, I think, really strongly argues for the importance of conservation now while we can. So with that, happy Earth Week, and I'll leave the what's next for later, and I'm happy to answer any questions. Here.
You talk about generating new niches.
Yes.
But isn't it part of the evolution of competitive exclusion? It's not like, the physical habitats are there. You just need to.
Exactly, but you
So it's not that the new niches are developing biodiversity. But the biodiversity evolved because of the competition as evolution occurred.
Yes, but if you have, so you have one new species that evolves and occupies a new, like opens up a new niche space, basically, right? Within that niche, then you have a lot of more things that kind of come in behind it and diversify. So when you open up that, like the first thing to occupy, to colonize a new niche, opens that up for that group of organisms, which can then kind of come back and fill in. Because like you think about like a very, very broad sense, like mixed layer, tropical niche, right? Is dozens of species that live in that.
But I would argue that you don't open new niches, but the competitive exclusion forces you to look for different niches, which is a different, egg and
right.
Chicken and
I mean, that's exactly,
So if I may just continue on that, what about the rate of just mutation, which is another thing that, what? I will embarrass.
No.
I mean, where is that here? Because the follow-up is that rate is to some extent
absolutely.
Determine the rate of biodiversity, which, in my opinion, determine the rate of need for new niches. I would call it a need.
Right. I mean, so the first part of what you said, I agree. It's a chicken or the egg thing, right? And is it competitive exclusion forcing new niches, or is it mutation happening to open up a new niche? And I think that it's always a cop out to say it's a little bit of both, but I do think that that's probably part of it. I am not exactly sure of the genetic mutation rate of foraminifera. There's been a lot of genetic work done, and I need to read more of those papers. But I do know that speciation can occur very quickly in forams. I mean, within the first 30,000 years after the impact, you had 20 new species appear. Basically, I mean, within a lot of them within like 10,000 years of the impact, very, very quickly. So it's not that speciation is kind of a slow process. Like that potential is there. It's just that there's either nowhere for them to go or no need for them to do that, if that makes sense.
Well, is it? I mean, even though you can have speciation occur at a varying rate.
Right.
But is there a time period necessary to increase the diversity just on mutation alone? In other words, is there a threshold value that you get enough diversity to create selection and then competition that creates niches? So in other words, the other way is the opposite driven.
Yeah.
So the niches aren't there until you have the diversity and the creation of competition.
Oh, definitely. Well, I think that's the thing is species create niches for themselves. It's not like an environmental niche, right? So yeah, so you have to have that spark of evolution to actually open up that niche in the first place. So I think it's an iterative process that opens up,
And one thing related that you didn't mention, what about just biomass? I mean, the overall abundance question of things. Even if you have a few species, because your abundance equal to diverse times when there's increased diversity. In other words,
yes.
Overall biomass.
Yeah. That's something I didn't have time to go into in this talk, but there's been a lot of work done on just the recovery of the number of foraminifera. Or the number of nanoplankton within a specific site before and after the KPG. Like I said before, at this particular site, the Cretaceous was vaporized, so I can't make that comparison. But people have done work, Chatsky Rise, and Walvis Ridge, these different like North and South Atlantic, North Pacific sites, where they've looked at basically just how long does it take to get to the same number of fossil foraminifera per gram of sample, and it's a couple 100,000 years. It comes back very fast. Same thing with nanofossils. So the biomass is there. You just have a much lower diversity assemblage. And to get back to your other question about genetics, I think that's actually a really interesting idea for a follow-up study on this. Is that so? If this is what's driving it, how do you compare this with random genetic mutation? And that data probably exists because people have done a lot of work on, like I said, on foram genomics. So I bet we could look at genetic mutation rates for like later in the Cenozoic. No.
There's no forum genomics.
Or yeah.
There's a problem. We have only one naked foraminifera. We actually don't have a genome because they eat all the junk and we don't know how.
That's true.
Yeah. We're trying. We actually will be trying now to do that.
Okay, so once we have that data, then that'd be a great hypothesis to test with it. Yeah.
Hi, Murray.
Yeah.
Yes, hello. So
Yes.
I think you have very, very precise don't think it will 10 million years for life to recover on the planet.
Mm-hmm.
So you have two modes. You have the mode of the successive species, as you show.
Yes.
And then there's the mode which are about 3-4 million years after the Big Bang, in which you start radiations.
Yes.
This is a completely different process because this is really when you open the new niche and indeed extinct, right?
Yes.
So I would not say if you didn't have the Paleocene boundary event and you kept going, then you would say, okay, it takes until maybe 12-15 million years longer to get full recovery of species, right?
Yes. So what I would actually, so I think that I kind of cut off a little bit for time purposes, but so the final recovery of diversity of foraminifera is, I mean, it's even off this plot. Yeah, it's like you said, it's like the middle Eocene is when you get that peak. I think nanofossils too, right?
You are mixing a lot of things here.
Okay.
You cannot understand the evolution of groups just on the number of species.
Oh, totally agree.
Okay?
Totally agree.
You have to look at the lineages, you have to look at others, at families, you have to establish your hierarchy. When you establish your hierarchy, I would say that by 4 million years after the Big Bang, you have a stable environment in which new lineages, stable lineage can develop. Before it's more difficult, but 5 million years, not 10 million years.
I totally agree, and I should be specific that this is talking about the recovery of species-level diversity, specifically species-level diversity.
All right.
Yeah.
So this is a real subject, and I'd love to discuss this with you afterwards.
Okay.
How do you recognize biology species based on paleontological data? You cannot, particularly in the coconut form.
Right. Well, with foraminifera, we use shapes as a proxy, but yeah, it's a proxy. It's not a direct. Yeah, well, like everything.
All right, I think we can continue that into a plot.
Sure.
So what's the line of reasoning that the Chicxulub crater, ground zero, should have a memory that's beyond overturned time of the ocean, that you expect anything different there than any other place? I mean, what's special about the recovery of Chicxulub?
So,
What's the memory?
so there's people have hypothesized that there'd be enrichments in toxic metals, things like that, for hundreds of thousands of years. Which longer, I agree. I was skeptical of this, but it was a hypothesis was put out there. The other big thing is the hydrothermal system, which I know that Sony has been working on a lot. But there is a hydrothermal system that you have with any impact crater, because you create a lot of porosity in the rocks. You have a lot of deeper rocks, deeper in the crust coming up, so you have a big source of heat. So you're circulating this water through. So you'd have
Nanos live on the surface of the water.
right, but there might be that might be a source of some sort of holding it back. These are I mean, yeah, I agree with you. I came into this skeptical that any of this could be a process, but there's a lot of hypotheses about that before we drilled there. So to go look at it and actually see was important. Yeah.
Is there any biological activity associated with hydrothermal?
There is. So they've found. Well, there's a paper that's working its way through reviews that has modern deep biosphere stuff, but I don't know about the hydrothermal system. I'm not sure if they found. That part of the science party hasn't been very communicative.
It's like Marco and Charles Cockle.
So I'm not sure if they have. They were
about
looking
the
for it.
modern species that are present in the crater floor, but they haven't said that much
A
about
paleo hydrothermal environment, yeah.
Did you guys look for mercury and all involved in there? I mean, hydrothermal sometimes, from the
Yeah, we've got a bunch of like ICP-MES and XRF data, so I imagine that we would see. But I don't know that we've looked specifically.
I haven't looked for mercury myself. but
Yeah.
That's
But-
interesting.
worthwhile to talk with John Reinfeld there.
Yeah.
This is people talking.
Yeah.
Yeah, I bet that data already exists.
Yeah,
That's what I'm
it's
saying. I
probably
bet
already been measured, but not been like specifically looked
Yeah.
at. Yeah. All right. Any student questions? This could put you on the spot.
Yeah.
All right. With that, let's all thank you.